Hi, welcome to our lecture on muscle tissue and organization. Here we can see the inside structure of a muscle cell, and that's just part of what we want to get to know in this chapter. We're going to begin here with this idea that muscle has four basic properties. Now this will kind of add on to our earlier discussion about muscle in the tissues chapter, suggesting that muscle can contract. Definitely this is true. We're going to recognize that as contractility. I've actually listed that second here because prior to contraction, the muscle cell must be excited. So we call this property excitability. And in this diagram here, we can see that we have a motor neuron sending a signal down here to our neuromuscular synapses, which will cause the excitation of this muscle. Now, in addition to the excitability and contractility, muscle has some extensibility. That is, that it's capable of being extended beyond its resting kind of form. If you want to figure this out, you can go ahead and put your arm out and notice that your arm is probably most comfortable slightly bent. That means that you have a little bit of contention coming from your biceps, but you can stretch it out actually by contracting your triceps on the opposite side of that joint, and that will straighten your arm out more so that it's fully extended. If you stop using tension from your triceps, you should kind of bounce back into your slightly flexed position. So we have this elasticity that bounces us back, as well as the extensibility, the ability to stretch. If we think about what we use muscles for, that would be our set of functions here. The most obvious, of course, is movement, and obviously this is going to be done in conjunction with our skeletal system. Our skeletal muscles will contract. For example, if we take a look here at the biceps brachii, we have origins here on the coracoid process and supraglenoid tubercle of the scapula. We have an insertion on the radial tuberosity of the radius. When we contract, we pull that radius closer towards our scapula, causing that flexion motion. We also use a lot of our muscles, abdominal muscles, muscles of the spine, to maintain our posture. We can also use some of these same muscles, for example our abdominal muscles, to support soft tissues. So this does provide a level of protection in areas where maybe we don't have the rib cage or the skull protecting underlying structures. Now down here we have our pyloric sphincter, which is a valve in between the stomach and the duodenum, the proximal section of small intestine. So these sphincters are circular rings of muscle that can regulate flow. Interestingly enough, this one right here is going to be composed of smooth muscle, but we do have skeletal muscle sphincters like this, our external anal sphincter and the external sphincter in our urinary system as well. Those are both going to be composed of skeletal muscle, giving us voluntary control over urination and defecation. And then, of course, muscles, when they contract, are going to utilize energy. As a result of that, they're going to produce heat, so we can use muscles to maintain our body temperature. All right, so we're going to begin here looking at muscles by thinking about the connective tissue that wraps them up. Muscles and nerves have this same basic pattern of this. It's just that where we say epimysium here, we would say epineurium if we were talking about nerves. But both of them are basically bundles of smaller units, and they are wrapped up in some kind of connective tissue wrapper. So the epimysium is this outer wrapper here on the outside of a whole skeletal muscle, and it's going to wrap together a bunch of structures that are smaller we call fascicles. So these fascicles are also going to be wrapped up and separated by what we call some perimysium. So the perimysium is going to be around a fascicle here. We've got it around a fascicle here, around a fascicle there. Each fascicle is going to be wrapped in perimysium. So again, if we think about this, epimysium around a whole muscle, perimysium is going to be around fascicles, and then we're going to get even deeper in here. Each muscle fiber, which is a synonym for a muscle cell, is going to be wrapped up by endomysium. So the endomysium is this layer here around this single muscle cell. We can see here that here is a single muscle cell or muscle fiber. There's another one, there's another one, there's another one. Each one of them has this endo, meaning inside, endomysium wrapped around it, but they are wrapped up in perimysium to form a muscle fascicle. and multiple fascicles are wrapped in epimysium to form an individual muscle. Now, we do, of course, get kind of a convergence of these connective tissue layers to give us these thicker pieces of connective tissue that do important things. So as we start thinking about the mesia binding together and forming something, they're going to form tendons, and they're going to come in a couple of different forms. Many of our tendons are going to be thick like this or this. So this is the tendon of the short head of the biceps brachii. This is the tendon of the long head of the biceps brachii. And they are cord-like, which is our standard form for tendons. Down here, we have our thoracolumbar aponeurosis. It's this whole white banded area. And an aponeurosis is a tendon. It's still a convergence of all of the mesia, and it's still going to connect a muscle to bone of some kind. But an aponeurosis is a flattened sheet rather than a kind of a thick cord. So you can imagine that this will be relatively thin if we were able to pinch this, but it does cover a very broad area and is great for attachment of a large muscle like the latissimus dorsi here. All right, so let's go ahead and get back and review those subunits that we had to talk about while we were talking about the various connective tissue wrappers or mesia. We're going to go ahead and begin with the idea that a fascicle is a bundle of muscle fibers. So a whole muscle is a bundle of fascicles. But once we see this whole muscle forming multiple bundles or fascicles here, each fascicle itself is actually a bundle of that smaller unit, which are going to be the muscle fibers or muscle cells. Now, even as we see an individual muscle fiber, it's clearly a bundle of smaller units. Those smaller units are referred to as myofibrils. And we can see a myofibril popping out right here. And then this is a portion of a myofibril, because we're going to recognize that myofibrils now are not bundles of something else, but rather more like a chain of something else. A myofibril is a chain or a set of connected sarcomeres. A sarcomere goes all the way here from this area we call a Z-disc, which is one end of it, to a Z-disc on this other side, and it's going to consist of various zones or bands. So we're going to have I bands, A bands, and an H zone or H band, and it's formed by thick and thin filaments, the thick ones being myosin, and then the thin ones being actin. So just as a reminder, to go back through this again, a whole muscle is a bundle of fascicles. A fascicle is a bundle of muscle fibers, which is a synonym for muscle cell. A muscle fiber is a bundle of myofibrils, and a myofibril is a big long chain of sarcomeres. Sarcomeres are not only the repeating unit within a myofibril, they are the unit of contraction. And as you can imagine, if we can contract a sarcomere, it's going to get shorter. If a myofibril is a chain of sarcomeres, it's going to get a lot shorter as each sarcomere inside of it contracts. And of course, when we contract a muscle cell, we can contract all of the myofibrils inside of it to give us strength. We can contract all of the muscle fibers in a fascicle should we choose to excite all of them. And we can use whatever number of fascicles within a muscle we need to in order to get the strength of contraction that we wish. So let's address kind of a weird question here before we move on. Why are these muscle cells so long? Obviously, muscle cells are long. They've got a bunch of myofibrils in them. Muscle cells start out as these myoblasts that differentiate into these satellite cells and immature muscle fibers. And we're going to get these immature muscle fibers here fusing together to form an incredibly long muscle fiber or muscle cell. So as we start kind of doing this immature muscle fiber gets bigger and bigger as these individual cells start to join to it. It also helps to explain why, of course, it would be multinucleate. All right, if we think about contraction, we don't always need the same strength of contraction, but we do have kind of a basic setup here to where we have muscle fibers working together in groups. All of the muscle fibers controlled by a single motor neuron are referred to as a motor unit. And you can see in this image here that we have three different motor neurons traveling to this particular fascicle, and we have a bunch of muscle fibers here broken up into 3 separate motor units. So how much strength do we need? Do we need to use a single motor unit? Do we need to use 2 out of those 3 motor units? Or should we use all 3 of them? Those are kind of the options we have, because either you can have a neuron go ahead and excite all of the muscle fibers that it connects to, in other words, use a whole motor unit, or you can not use that motor unit. What you can't do is use part of a motor unit. All right, so question, what does the motor unit size, the number of muscle fibers that are connected to a single neuron, tell us? And it tells us something really important. The fewer muscle fibers per neuron, the greater control we're going to have, because we can just use a few muscle fibers, or if we need to, we can use more than one motor unit to use lots of muscle fibers, but we have all of those options. Now, if we end up having hundreds of muscle fibers for a single neuron, our option is either to use 0 or hundreds of muscle fibers. So you can kind of imagine that it really matters what kind of control we have, dependent on the size of those motor units. If we have just a handful of muscle fibers connected to a single motor neuron, we can have a very small or kind of slight contraction. But if there's tons of them, it's only going to be a powerful contraction or no contraction. And we can think about this looking at this homunculus here of our primary motor cortex to kind of see how much brain has to control a particular area. Although we have some big areas like our knee and our hip, which have a relatively small amount of brain, we have an incredible amount of brain controlling our hand, which is a smaller, weaker area. But this area is going to have to send more neurons to it, because each neuron is going to connect to fewer muscle fibers because we have smaller motor units in areas like our hand, or going to our extrinsic eye muscles, the ones that move our eyeballs around. What this means is that we can have some very precise and delicate movements in these kinds of areas, whereas we can get away with these larger motor units, a single neuron connected to hundreds of muscle fibers, in places that move our knee or our hip, where we're going to need powerful contractions that are going to work against our body weight, and we don't need that kind of precise control. All right, so as we are thinking about exciting some muscle fibers, we're going to do that at what we refer to as a neuromuscular junction, or a synapse. Now, synapses occur in a variety of forms, so this neuromuscular junction is specific to this kind of connection. But a synapse is always going to be where we have communication through neurotransmitters, where one neuron is going to give information either to another neuron or to an effector of some kind, an effector being either a muscle or a gland. So we just refer to this as the site of chemical communication between a nerve, this would be a motor neuron, and a muscle fiber. And if we think about kind of the way this is going to work out, we're going to go ahead and make sure that we have some kind of nerve transmission. We refer to that as an action potential. We're going to release acetylcholine from the end of that neuron. This would be a synaptic knob, or we sometimes call these things telodendria. And as we release that acetylcholine in there, it's going to bind to receptors on the muscle cell. So this is in the sarcolemma, or the plasma membrane of that muscle cell, causing excitation in the muscle cell itself. This is going to result in a calcium release, and calcium is going to affect these protein fibers inside here, the thick and thin filaments that we were talking about earlier. So you can see kind of that we have in green here our actin, our thin filament, and this is our thick filament myosin. Myosin needs to connect to the actin, but we have a problem. We have something in the way. So what's going to happen is the calcium is going to bind to this troponin, and it's going to be able to actually move these bands, these other proteins, out of the way so that the myosin can bind to the actin, forming what we call a cross bridge. From there, we're going to use our ATP to change the shape of this myosin head to create what we call a power stroke, and that's going to cause the shortening of our muscles. Now, everything we just covered, you're going to cover in more detail in your physiology class So we're just going to worry about the anatomy. And we are really concerned about the sarcomere. It's kind of the key feature of this chapter. And the sarcomere consists of partially overlapping protein filaments. So it's going to have this kind of partial overlap, and we need to increase the overlap in order to get a contraction to take place. The two proteins we are most concerned with are actin and myosin. So actin in this particular area here is shown in red in this diagram. It's our thin filament. And then in kind of this blue or purplish, we have this myosin with those little heads kind of poking off the side. So this is kind of our overall structure. You can see here we have an area of overlap. We have another area that's not overlapping and another area that's not overlapping. We could say that this is thin filaments only. This is overlap. This is thick filaments only. And that information is going to help us divide this up into different sections. We'll begin first by recognizing that there has to be kind of a central anchorage and a place at the ends of a sarcomere where it connects to another sarcomere. So the M line is going to be the middle of this. The good letters right here. Every letter in here is good and helps us identify what part we're dealing with of the sarcomere. The Z lines have this kind of zigzag on the end. Of course, Z we also think about as being at the end of the alphabet, while M both stands for middle and is relatively close to the middle of the alphabet. So what we'll notice here, if this is the end of our sarcomere goes from Z line to Z line, but we have a sarcomere on either side of that as well. Now, as we get kind of deeper into this, we need to take a look at the various bands, and these are the bands that we can actually see when we see striations in muscle. We have our H band and our I band to take a look at in this particular image. The H band is in the middle. So we kind of see that here. You can see that it's only the area that has thick filaments only. It includes the M line, but it's not including the area of overlap. We have the I band, and the I band actually extends from our sarcomere over to the adjacent sarcomere because it's an area that has only thin filaments. Now, if we think about these, the I is a thin letter, thin filaments. The H is a thick letter. It has the thick filaments only. And then we have our A band. The A band is the region that's going to be in the middle, and it's going to have the overlap. If you take a look, the A is thin at the top, and it's thick at the bottom like an H. So that reminds us that it has both of those areas. Now, if we take a look at this animation down below here, we can kind of get an idea of what's happening when we have our contraction and how it relates to the sarcomere. We'll start with a relaxed sarcomere here, and we're going to have a contraction because we have calcium, those little things that are moving around, flooding in. We decrease the non-overlapping spaces, and in relaxation, we're going to go ahead and increase the non-overlapping spaces. Let's take a look at our image up top here. Up top, we have a relaxed sarcomere. You can see that I have some I band in here, and I've got all of this H band in here near that M line. And if I take a look at a contracted sarcomere down below here, I no longer have any H band. It's really disappeared, and it's disappeared because I've increased the amount of overlap. The overlap gets rid of those areas. Now the I band is not gone, but it has decreased in size. So a contracted sarcomere will really have no H band at all. It's going to disappear during the contraction as we increase the amount of overlap, as those myosin heads start pulling the actin towards the M line. And then when we relax, we're going to allow those actin fibers to move back again. That's actually using another protein we call titin, which tends to be elastic. All right, so finally, thinking about muscles themselves and getting ready to talk about and learn a bunch of individual muscles, we want to think about what do we need to know about an individual muscle to tell what it does. And we need to know basically its origin and insertion. The origin and insertion are both attachment sites, and so sometimes there's gray area where an origin isn't obvious and an insertion isn't obvious because you can't tell which attachment site is which. But most of the time, we're going to find that origins, like in this case, the coracoid process and supraglenoid tubercle of the scapula, are going to remain stationary during the contraction. And down on the other end, in this case the radial tuberosity, being the insertion of the biceps brachii, that's the insertion because it's going to move during the contraction. In other words, what we can say here is that as I contract my biceps brachii, I'm going to pull the radius closer to the scapula. And that's generally what's going to happen. When we learn origins and insertions, you want to think about the origin as staying still and the insertion as being pulled towards the origin. That is the action. So if we think about our biceps, go ahead and extend your arm, contract your biceps. You're going to notice that you are flexing your antebrachium. Whenever we talk about an action, we're going to use that combination of terms. Flexion is the term I use to indicate what kind of movement is taking place. I am decreasing the angle in those joints. And the antebrachium is the thing that is being moved. Now, oftentimes we could actually use the name of the bone. So I can't flex my radius though because the ulna is strapped to it through a syndesmosis and they behave as a single unit. So it would be most appropriate to say in that case that I'm going to flex my antebrachium. Now that takes us to some basic terminology because we know that muscles are going to perform particular movements, and some muscle, the agonist or what we call prime mover, is going to be the muscle whose contraction is mostly responsible for a particular movement. Now this isn't always super easy to figure out, but here's a nice example here. This is the spine of the scapula going across there. That means that this is the infraspinous fossa, and the infraspinatus muscle originates in that, and then it's going to insert on the greater tubercle of the humerus. It is the prime mover or agonist for lateral rotation of the humerus. So pretty simple thing to kind of think about there. It's the biggest muscle that causes that lateral rotation. It is assisted by smaller muscles like this one down below here, which is called the teres minor. We also then have what we refer to as synergists. If the infraspinatus is the prime mover of lateral rotation of the humerus, the teres minor is a synergist, as is the supraspinatus. So we could think about all three of these as working synergistically. And to be honest, for us, this is maybe the most important term, or one of the two important terms, because this tells us that we have groups of muscles that do the same job, and we can basically learn them together. Now the opposite of that is to be an antagonist, and here we have our great antagonists pair of our brachium. Now this biceps brachii is going to lead to flexion at the elbow or flexion of the antebrachium. Either way you want to talk about it, movement at that joint or movement of a particular body part is perfectly good. It actually has some synergists. The brachialis and the brachioradialis are synergistic with the biceps brachii because they both also flex the antebrachium. But on the opposite side of this region is the triceps brachii, and it's going to go from attachment sites on the posterior humerus as well as the infraglenoid tubercle of the scapula to insert on the olecranon process of the ulna. If you are flexed, it pulls that olecranon process backwards towards the humerus, extending the antebrachium. That's exactly the opposite movement of the biceps brachii. So the biceps brachii and the triceps brachii are antagonists.